Here we go. End of the unit. This is going to be the last portion of Subtask 2. Again, in the learning targets, you're going to find these on page 8 and 9. This is going to kind of wrap up the end of the story with DNA replication. So, as we've talked about, DNA replication can only occur in a direction. It has to go from 5' to 3', meaning that that nucleotide can be connected to the one below it from carbon 5 and 3 being connected with a phosphodiester bond. You could not take a nucleotide and put it on top of this one. That would be building in the wrong direction. Now, there's a little bit of a secret here. As all these unassembled nucleotides are floating around, they actually don't exactly look like this. In fact, it's worth noting that putting them together from 5 to 3 by enzymes like DNA polymerase, that's going to take some energy. And the energy in this case does not actually come from ATP. You'd think so, because ATP is our energetic molecule and it seems to power everything in cells, but I'll remind you that ATP is a nucleic acid. In fact, it looks a heck of a lot like a DNA nucleotide with its nitrogenous base, its ribose sugar, and a phosphate. It just happens to have a couple more. Well, it turns out that nucleotides, before they are added, are actually deoxynucleoside triphosphates. It's a nucleotide, like we've been drawing, but it has 2 extra phosphates on it. Those phosphates store energy, and so as each nucleotide is added, the 2 phosphates are lost, and that provides the energy to the enzyme proteins to do this work building. So the secret here is that nucleotides don't actually always look like this. Before they are added, they look like that. They have a slightly different name, deoxynucleoside triphosphate, and they act as their own ATP. Now, the next thing that we'll look at is the way we actually end up putting these together. So we have this shape. There's the 2 parent strands, they get separated, and we've noted that the new daughter strands have to be built a little differently. One is a leading strand, one is a lagging strand. But that's not the whole story. It turns out that replication forks actually exist on 2 ends. A replication fork up here, and if you follow it, there's more DNA and another replication fork down here. These 2 replication forks started together in the middle here and are moving apart from each other as the helicases unzip them. This creates a bubble. And it turns out that's not the whole story, because if you only had 2 helicases running in opposite directions, it would take way too long for you to copy all of your DNA in a reasonable amount of time. So in eukaryotes like you and me, our chromosomes are actually going to have a bunch of different bubbles happening. Each bubble has 2 forks that are running away from each other, and in this case, there are so many places that replication is happening at the same time. Literally, if I were to mark it, replication is happening here, 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 all over. So there's so many sets of these enzymes working at the same time, you can actually do your replication quite a bit faster. On one strand of DNA, there are going to be a whole bunch of different places where replication starts, the forks open up and start running away from each other. Here, these 2 forks are about to touch each other and they'll just form a larger bubble once they meet. So, at this point, taking a look at that whole image with all of its individual replication forks and bubbles, I want to give you a chance to kind of break that down with a simpler visual. So here's what I would like you to imagine. Right here is an origin of replication. That little dot represents the place where a set of enzymes come in and start replication, breaking the DNA apart and forming 2 forks that start to spread. Here's 2 more forks, here's 2 more forks. Here they are a little bit later on. They've had some time to spread out. You can see that fork and that fork are about to run into each other. The 2 helicases are driving towards one another. after all of these forks have met and the bubbles have completely opened up, you have two finished DNA molecules. So the initiation points, the places where replication started, if you could rewind this and see where the forks began, that is known as an origin of replication. And I'd actually like you to use that word at the beginning of Subtask 2 in that first essay. Proteins called origin recognition complexes bind to these origins of replication, and that is what allows DNA helicase to begin its unzipping. So replication forks move away from the origin in two directions until it hits another replication fork racing towards it. Now, the last thing to talk about is the difference between how replication works in prokaryotes and eukaryotes. I'll remind you that prokaryotes, or bacteria, have DNA that is fundamentally different than ours. It's a loop. And so when you have a loop of DNA, you actually don't need a whole bunch of different forks in different places and a whole bunch of bubbles happening all along the chromosome. You only have one chromosome, and it's a circle. So what you can see here is a real photograph of replication happening. Here's an artist's interpretation of what you are seeing. Here is a fork, here is a fork, there is one open bubble, and eventually those forks are going to meet up on the other side, and replication is finished. So replication in prokaryotes involves replicating a single looped DNA chromosome. The replication is bidirectional. It only occurs in two directions with two forks. There is one origin of replication. If I were to imagine these forks sort of zipping back up, I could imagine that the origin would be right here and right here, where they used to be together, and that's probably where it started, with the forks then racing away from that point. So in a prokaryote, there is only one origin of replication, there are only two replication forks, and it makes one growing bubble until that bubble separates the entire chromosome into two identical circles of prokaryotic DNA. I'll also remind you that this DNA is not packed up, it's not supercoiled. There are no histone proteins or nucleosomes, which is why we call prokaryote DNA naked DNA. Prokaryotes have naked DNA, we have histone proteins. And that, folks, is the end.